Heather Wakely, Deputy Director of the Stanford Cancer Institute and Chief of the Division of Medical Oncology. And today's breakthrough seminar is going to be featuring Dr. John Heymach, who I have known Dr. Heymach for almost probably my entire career, because we've had very parallel paths in some ways, working in the field of lung cancer. And so this man has made incredible contributions across lung cancer and more broadly thinking about targeted therapy. And before we knew about targeted therapy, really focused on angiogenesis, and now coming back to that as well, as all things sort of intersect. But John is actually a New York native, but then went up to Harvard for his undergraduate degree before coming to the farm for his MD, PhD. So he spent a number of years with us. He then moved back to Boston and did his internship and residency at the Brigham Women's Hospital, fellowship in medical oncology with Dana-Farber and Mass General. So regardless of how all those three have merged and unmerged and remerged, he was with all of them at some point during that training time. And as a physician scientist, Dr. Heymach's research focuses on investigating mechanisms of therapeutic resistance to targeted therapies, in addition to understanding targeted therapies before there is resistance. A lot of his earliest work was in angiogenesis back in the time when we thought we were going to be able to figure out the biomarkers there and make progress there, but really some very impactful work in the field of angiogenesis as well. And then thinking about biomarkers for targeted therapy as well as for immune therapy, we were talking a lot about our mutual interest in brain metastases as well, and other really striking therapeutic challenges that we face as lung cancer clinicians. His work has led to really impactful changes in treatment of KRAS mutant lung cancer, EGFR lung cancer, small cell lung cancer, adenoid cystic carcinoma as well, branching out a bit. And then a lot of our recent work for both of us has been in earlier stages of lung cancer and sort of bringing some of those advances forward there. He is the PI for 4 R01s. That's where we diverge. I am not the PI on 4 R01s. And they're all focused on molecular subsets of lung cancer. There's a U01 focused in small cell. He is the PI for the Stanford Cancer ACS Lung Cancer Dream Team, which is focusing on KRAS work at MD Anderson. Also leads the lung CCSG program, co-leader of the lung cancer moonshot. He is the PI and director of the lung spore as well. So the fact that he was willing to come and spend the last 2 days with us is truly phenomenal. So thank you. And as a clinical investigator, he's led numerous, I can't even count them, number of clinical trials. He has also sent many of his children here to Stanford, so we appreciate that very much. And that has allowed him to come and visit us on a regular basis. One of his daughters is here today. I will call you out if you want me to or not. So, and I'm really looking forward to the next hour and hearing your talk on Beyond Driver Oncogenes, New Paradigms for Personalizing Lung Cancer Therapies. So welcome to Dr. Heymach.
Okay, well, thank you so much, Heather. And it really is a pleasure for just a lot of reasons to be here. And, Heather and Joel and the lung cancer group have been collaborators and Heather and Joel friends for a really long time. And, I've been reminded just about a part of the special sauce of being here at Stanford, the thrill of just talking about cool science, and just today getting to talk with, Nathaniel and Peter and Stephen and then finally getting to talk with Michelle, who we've been interacting from a distance for a long time. It just reminds me about the excitement that happens here that you all really should value. It really does make the place special, of just talking about cool things that are scientific wherever they may lead. And that's really part of what I appreciate here. So I'm going to talk about Beyond Driver Oncogenes, new paradigms for personalizing lung cancer therapies. And this comes from the fact that, I live with one foot in the clinic. What I and hopefully our group does is really try to say, the problems that we have in the clinic, how can we really focus on tackling those with the scientific skills that we have. And let me just briefly tell everybody how I've gotten back to Palo Alto here. So as Heather mentioned, I did chemistry originally. So my PhD was in neuroscience. So a couple hundred yards from here. So the Fairchild building, second floor, right behind us. So there's Eric Shooter, who is my mentor. Barbara Barris at the time, later Ben Barris, was also a mentor, and we did a lot of work together. This is his wife, Elaine. Unfortunately, they've both passed away recently, but he's really a wonderful scientist and a gentleman there with my wife. And so there was work on neurotrophins. And at the time, our group identified that neurotrophins, where the receptors were just being identified and the TRAC receptors emerged, we identified that they were tyrosine kinase receptors. Later, Mariano Barbacid identified the TRACs themselves as the receptors. And, a lot of this was how neurotrophins get sorted and secreted and regulated. And, so I began to think that signal transduction was really a way that we could intervene. So I decided that was really something I wanted to pursue in more depth. But I always realized doing it in the CNS was really hard. I wrote a lot of grants about how we were going to treat Parkinson's and Alzheimer's, I came to learn that your brain is surrounded by this hard skull, and it's really hard to get through clinically. And, by the time neurons die, it's pretty late, to intervene. And so, as I looked ahead, I thought, let me try to tackle something a little bit easier. And so that was lung cancer. And so that, I guess, has worked out, reasonably. On a relative scale, it's been somewhat easier. And so then I went to work with Judah Folkman in Boston, and the focus there was VEGF receptor inhibitors and angiogenesis. Again, similar signal transduction pathways. And that's where I sort of cut my teeth on tumor microenvironment, angiogenesis, a little bit on the immune system, and about how to target RTKs. Now, the time here at Stanford was also productive for other reasons. Here's my wife with daughters Julia and Claudia. Claudia was born, in Packard. And Claudia with her cousin Rachel, the blonde-haired one there. And Claudia and Rachel, class of 2018. So, and Julia seated over there as well. What we think about, and I think the key to personalizing therapy is understanding heterogeneity. And, heterogeneity means a lot of different things. I see an infinite number of single-cell papers that say, look, we found heterogeneity. And I've never seen anybody say, wow, we found no heterogeneity. So I think the term gets sort of washed out a bit. So let me talk about how I really think about it. We're focused on, when we see a heterogeneous disease, are there therapeutically tractable subgroups that we can do something about? I don't care particularly how we get to those subgroups, but the guiding principle is, are they tractable therapeutically? Is there some different therapy we'd give as a result of looking at these subgroups? Now, we also deal with intratumoral heterogeneity, and this is our enemy that we all know that, tumors become more heterogeneous as they go along and they become harder and harder to treat. So we do have a focus on that. But that doesn't guide our initial therapies yet. Really, it's the first one that's been the focus of what we've done. And I think we've developed habits of looking at a given disease space and saying, where's the heterogeneity, and is it heterogeneity that we can do something about, however we can sort it out. Now, the chemotherapy era, I started treating lung cancer, now more than 2 decades ago, like Heather, we're contemporaries. And, when we both started, lung cancer was defined histologically, and it was pretty straightforward. I think that may have been part of the decision. You could learn it in about 45 minutes. You had looked under the microscope, and my mentors literally looked under the microscope, Art Scaron and George Canellis and others, and said, that's a squamous. And then, our therapeutic choices were chemo, and chemo. And, we had lots of debates about what we saw under the microscope. None of it made a difference. So that was it for a long time. And I think a lot of people identify this study by Joan Schiller as for lung cancer when everybody realized we just had hit the ceiling. This was comparing our leading 4 chemo regimens and they were all terrible, and they were all equally terrible. There was no difference between them, despite the fact that there were avid fans of gemcitabine, avid fans of taxanes, and at the end, they all had a median survival of 7.9 months, objective response rate of 19%. There were studies that said triplets were were not better than doublets. They were more toxic than doublets. Doublets were better than singlets. So that meant you were stuck at doublets, and they were all equally bad. And so I think a lot of us said, well, okay, we're either stuck here and go move on to something else or find different approaches. Now, back in the early 2000s, so, our group and efforts led by Matthew Meyerson and Pasiani and Bruce Johnson and others, our group first identified EGFR activating mutations. And I think that was really what we see as the beginning of targeted therapy for lung cancer. Now, what was progress like during this chemo era? Well, you see an upward curve, but if you can read the axes there, over 25 years, median survival of lung cancer went from 6.9 to 7.3 months. All right, so you don't need a lot of advanced math to say that over 2 decades, less than a half a month difference is not the sort of progress we want to be making. So what we've really tried to do is say, here's a heterogeneous population. We have different drugs that may be highly effective for one or the other of this, and can we find the heterogeneity and detangle it? And this is where our biomarkers come out. So can we sort these out? And then once we've sorted these out, we have confidence that we'll be able to find better therapies for each of these different subgroups. So we really see detangling this interpatient heterogeneity as a necessary first step, for developing a more tailored and effective therapies. Okay. So what does the current landscape look like of lung cancer? Well, nothing like it did during the chemo era. We have what we think of as the driver negative group. It's a little more than half. There we really tailor therapies right now predominantly on just PD-L1 levels. And the driver positive group, and coming close to half depending on which population you're looking at. And so we now have 11 different oncogenes that we need to have information for before we can treat anybody with advanced disease. And in fact, if you're a medical oncologist, a lot of the decisions you make no difference whatsoever. The one thing that makes an absolutely huge difference is if you find a driver oncogene when it's really there. Missing a driver oncogene is the one decision a medical oncologist can make that can lose a decade or more of life very easily. So this really is the first branch in our hierarchical tree of decision making. Now out there we also have small cell, and we'll talk a little bit about that at the end. Okay, so what's the sort of improvement now for those here who don't treat lung cancer? Well, I showed you over 20 plus years, it was less than half a month of progress. The lung cancer docs here very well know the CROWN study. This is the randomized phase 3 with lorlatinib. The median progression-free survival is more than 5 years here from just taking an oral medication that's reasonably well tolerated. All right, so same patients on the left and the right, and now here's a subgroup. Their median overall survival is north of 8 years now. So coming up on a decade of survival just by identification of a driver subgroup, and then wonderful pharmacology from people like Nathaniel and others. Okay, so that's worked really well. The case with the CROWN study is awesome. So can't we just turn the crank on that and do a lot more of it? So I think the theme of my talk is probably not. I think that crank can only be turned so much. So the first is we've likely identified almost all simple druggable driver oncogenes in lung cancer to a frequency down of 0.1 to 2%. So we have pretty good confidence that drivers that are out there are below that frequency. There are rare fusions we come across from time to time. So there are no big slices in all likelihood. But the other factor, and I'm going to spend the first half of the talk about this, within an oncogene, there may be many different mutations, right? So, for the first 15 years of treating lung cancer, We just thought about classical EGFR mutants, the L858R and the exon 19 deletion, but there's over 100 different EGFR mutations. So now the next generation of questions is, can we figure out how to tailor for a given oncogene in the landscape of heterogeneous mutations? And there's additional sources of heterogeneity that we have an increasing appreciation for. So, for example, heterogeneity can be driven by the presence of tumor suppressors like P53, STK11, KEAP, and so forth. Transcriptional programs driving lineage, some of which may have to do with cell of origin. And this is, work that, Julian here and others have done a lot of work on. Where does the cancer cell start and how does that impact its future trajectory? Epigenetic modifications, and then a general category of stuff we don't know yet. So this is saying that I think, the good times that we've had by thinking that driver oncogenes would get us where we want to go, I think we're dissuaded of that, and it's gotten us very far down the road, but not where we ultimately want to get to yet. So we really are going to need novel approaches to get better at tailoring therapies more effective. So I want to give one example here of one approach we've taken to trying to more effectively tailor targeted therapies. And this is the case of atypical EGFR mutations. And I just want to acknowledge our EGFR team. Shining and Yasser are part of our clinical team that does a wonderful job. And in the lab, Monique, Jackie, and Yuji Shibata have led a lot of the preclinical work I'm going to show. So this is a list of recurrent EGFR mutations that occur in more than 0.1% of the population. So more than 0.1% of the EGFR mutations. So, this is something like 100 different mutations. Now, we've colored in gray here where there's explicitly an EGFR TKI approved for those mutations. And you see the vast majority of mutations don't have a TKI explicitly approved. I say explicitly approved because there's a blanket general label out there for afatinib. But so, what are we going to do with this huge number of mutations? Well, the first thought may be just take our best drug and give it to everybody. So our most widely used TKI is called osimertinib. It's a good drug. It's well tolerated. The median progression-free survival, if you just take osimertinib by itself and you've got inactivating classical EGFR mutations, about 19 months. That's pretty good. Well, here we've shown atypical mutations versus typical mutations for patients getting osimertinib. And you can see for classical, from our data set, it goes north of 20 months. Atypical mutations, it's 6.4 months. So if we just try to stamp a one-size-fits-all approach for EGFR mutations, the results are not great. So there's significant heterogeneity. And one thing we like to think about in the decision of splitting versus lumping, there's scientific considerations, but there's also practical considerations. And those of us, that do clinical studies, and I see a number here, Millie and Joel and Heather and many others, you have to have a population that's big enough to study. So we can't do 100 different clinical trials for 100 different patients. And it means that we were getting patients showing up every day that had no information about what the best drug was. So we felt this would be a problem we could tackle. So, we give some thoughts. So what's the best approach to classification in general? And, if I gave you a bunch of objects, an ant, a German shepherd, a cat, an alpaca, a Bugatti Veyron, Corvette, airplane, and so forth, and you said, well, how should we organize all these objects? Well, the simplest way is, just put them alphabetically, A, B, C, and go down the list. And this is great if it's the index of a book. But what if you're trying to say, the kids really want a pet, what should we get, right? This isn't the best thing to start with A, or what are we going to take to, move our stuff to a new state? So this isn't a one size fits all. A second algorithmic and straightforward approach is to do it by frequency. You can just count them, right? So this is deterministic. You can do it precisely. There's used to be 450 Bugatti Veyrons. Various Silicon Valley moguls are crashing them at some rate, so that number is going down. They're not making any more. So that number is still close. There's over a quadrillion ants. So you just count them and line them up by frequency. Well, does that help us with stuff? Well, you can classify them unequivocally, but I'm not sure what it helps you with. Or you could sort of organize them by what they do into pests, pets, transport, and sports cars. And, so, the answer to your midlife crisis, probably can be answered more easily by this sort of organization. So how have we done this for EGFR mutations? We've got over 100 EGFR mutations. Well, surely we took a really rational approach. Well, we took the counting approach, the frequentist approach. We literally, this just shows the frequency of each of the mutations. And, we took the top couple and we called them classical or typical. The next three were covered by the afatinib label because it was the three that they could detect at a high enough frequency. And so we ended up calling those the afatinib group. So that's one approach, and I think you probably get the drift that probably isn't the optimal approach for tailoring therapies. A second approach is to just use the exon, and there are trials out there that specifically tailor exon 18 mutations or exon 20 mutations. But that, of course, is just the location on a linear strand of DNA. So if our goal is to match mutations with drugs, why would we base it on their frequency or their exon location? So Jackie and our group led a big effort, and with more than 50 or 60 different TKIs, including a lot we made in-house, and across all the common mutations, we screened the drug sensitivity as using BAF3s. We've done other models as well, and we just started organizing them and looking at patterns of drug sensitivity. And here we've clustered them by sort of the generation of drug there. And you can look at exons, exon 18, 19, 20, and you see lots of blue, meaning sensitive, and lots of red, meaning resistant. And there isn't a particular structure to this data. In fact, we can do a Monte Carlo simulation and randomly pick a drug and ask, does knowing its exon give you better than random information? And the answer is no, with the exception of one small region of exon 20, the loop. So essentially, the exon doesn't carry useful information with a tiny exception there. And just to give you an example, within exon 20, showing the different generations of drugs, you can see wild variation just within exon 20 itself. So we developed, and this is using in part, clustering and AI, driven approaches, a structure function classification here. And we divide this into 4 groups, classical-like. So the red dots indicate where the mutations are. And if you're not used to looking at this pocket, this is the ATP binding pocket of the EGFR kinase. You would think of it as going feet first deep into that pocket. Green is where the gatekeeper mutation is, the T790. Blue is the P loop, and that's an interesting region, mainly in exon 18, where we get a lot of mutations like the 719 mutations there, for example. And one of the things with the red dots is that you see they're all around the pocket. They tend to open up the pocket, create one more favorable, for binding. And third-generation drugs work best for the classical mutations. We have T790, like these are deep in the hydrophobic core, different mutations that have a similar impact as T790 itself. We learned that there was a third group that was entirely unanticipated, and we called these the PAC mutations, standing for P-loop and alpha-C helix compressing. That's because, it took this alpha-C helix and it sort of pushed it in and pushed the P-loop down a bit. And again, we're lung cancer researchers, not marketers, as you can probably tell. But all of these different mutations had a similar effect and a similar impact on drug binding. Interestingly, for this entire group, our best drugs for classical mutations, third generation, worked much worse. And actually, older drugs, like our second generation drugs like afatinib, actually worked better. And then exon 20 loop insertions. These were insertions in a particular region of that loop that really caused a more dramatic steric hindrance on the binding pocket. So we asked the question of whether this would be a better way to classify EGFR mutations for the purpose of testing them and for patients. And the thought was that if this organization worked, well, then people would get better at drugging a specific structural group. We we had some leads, but we knew the world would be much better at drugging these if they knew exactly which ones they were going after. So here we're organizing it based on exon-based groups or structure function. You can already see there's greater order in a structure function-based classification. And we can show this mathematically, by doing a similar sort of Monte Carlo simulation or looking at the correlation between your structure group and drug sensitivity. And it turns out statistically it's a far better predictor there. So let me talk about that third group, the EGFR PAC mutations. This is one, again, that was entirely unanticipated. And this is one where second-generation drugs really were considerably better. Now, what these mutations did is they pushed down that P-loop, and it turns out there's a residue there, the 723 of phenylalanine, that has a pi-stacking interactions with the ring that occurs in osimertinib and third-generation drugs that turns out to be a very important interaction. And so when this loop gets pushed down, you lose that critical interaction for osimertinib. The second-generation drugs like the quinazolines didn't depend on that interaction there. Here, and this shows in a little bit more detail that F723 is the key thing that we think gets rotated down with these mutations. And we could show, even though osimertinib was far better for classical mutations, drugs like afatinib and poziotinib were much better for these PAC mutations. PAC mutations are a pretty big group. They're bigger than exon 20 insertions. They're about 12% of all lung cancers. Now remember, EGFR mutant lung cancer is about 40% of lung cancers in Asia. It's about 20% in the US. Here at Stanford, probably 30, 35 or so there. And we could show, if here we look at time to treatment failure on afatinib, time to treatment failure with the PAC mutants, second-generation drugs like afatinib did better than first or third-generation drugs, but it didn't apply to other non-PAC mutations in terms of that improvement. Now, that's great that we found a group that afatinib works better for, but for those of us, and I suspect there's only 3 or 4 in the room who have ever given afatinib, it's not a great drug. It's got a lot of toxicity. It doesn't have any CNS penetration, causes a lot of wild-type EGFR rash, diarrhea, and so forth. So the question is, can we come up with better drugs now that we've got a subgroup to go after? So we've done a lot of drug screening. We've developed drugs ourselves. I think the one that we identified in the clinic that looked really good was one called furmonertinib, or Firmo for short. There's a trifluoroethoxy group that we've got circled there with the arrow that has some really key interactions that osimertinib doesn't have that we think contribute to the improved potency for PAC mutations for Firmo. And we could show it worked well in preclinical models for PAC mutations. So Aravent opened a clinical study based on this new classification with Firmo at different doses. And really high response rate was seen, about 68% response rate. We think osimertinib for this group is in the neighborhood of 30 to 40 with a relatively short PFS, depending on the data set you look at. So now by identifying a group, we could repurpose drugs that sort of matched it really well. So based on this, now Aravent is doing the registrational study that has just opened called the Alpaca study. So this is a global randomized phase 3, and if the study's positive, it'll be the first new drug approved for PAC mutations. And we believe for this entire group of mutations, by giving it an identity and a structural basis, companies and investigators will be much better at developing drugs for it there as well. And there's now a number of other TKIs that are going after this subgroup that emerged from our study here with sutetinib, a Black Diamond compound, or a compound, and others. And now a number of different molecular diagnostic reports are reporting this classification as well. All right, now another subgroup to highlight here, and I think you'll see some similarities with the study, is exon 20 loop insertions. Now, what drove this is we had a faculty member that actually, whose wife had an exon 20 loop insertion, and we really spent time thinking hard about this and going back to the lab. And the mother of one of my daughter's dearest friends. So suddenly this, it became clear to us how poor our choices were, back then, it was about 7 or 8 years ago. Now, about exon 20 insertions, they occur not just in lung cancer, but more than 20 different tumor types. And the non-lung ones are more numerous than the lung ones. They're just more spread out. And here we're showing for first and second generation drugs. The black line shows how standard mutations, classical mutations, do with first and second generation drugs. The red line shows how exon 20 insertions do with first and second generation drugs. You can't really have a PFS shorter than 2 months because you do your first scan at 2 months. So that is synonymous with being completely resistant, with one particular exception. And here there's really a more dramatic steric hindrance. Here we're showing how far the P-loop gets pushed down. So, we studied the properties that we thought would make a good molecule and did a lot of screening. And again, we were able to develop some compounds that we thought looked reasonable, but we had actually found one that had been previously studied and discarded. So this had been studied as a classic EGFR inhibitor, poziotinib, but we thought it was actually a good fit. We brought it into models and it worked really well. So we called up the company, and the company was no longer aware that they had it because leadership had changed and they had discarded the drug, but there was actually a big batch that had not been discarded yet. And we showed preclinically it worked well. We got them to start studying this clinically. And, we studied, these are all different exon 20 insertions. So even within this one little region of EGFR, you see there's a lot of different variations. And, you can divide this loop into near loop and far loop in gray and red, and then the helical mutants in blue. The helical mutants, by the way, are sensitive to anything. They're the easiest ones to inhibit. But, we were really dealing with the gray and the red ones there. And one of the things we saw is that the location in the exon 20 actually made a big difference. So here we're plotting the location as you move along the exon, and then the IC50. So the higher the dot, the less potent the drug is against that. And so you can see the poziotinib got worse as you went further. Interestingly, mobocertinib actually got better for far loop than near loop. So you could imagine as you get new drugs, you may tailor them not only for exon 20, but where in the exon 20 loop insertions that they might be happening. And, doing molecular dynamics simulations is a collaboration with John Lawson at NASA and with our modeling group. I think we came to understand some of the factors. One of them is that the reactive acrylamide group that covalently links with the cysteine C797 equivalent got much further apart with the far loop mutation. It was much harder for this to covalently link, and some other considerations there. So we discussed with the company, and we recommended they only study poziotinib for near loop mutations. But they pointed out that studying near and far loop was a bigger market, and they would like a bigger market. So you can probably see where this is going. So we studied this for the overall exon 20 group. The response rate was 32%, but there was a lot of EGFR-related toxicity, diarrhea, rash, and so forth. Now, the bar by the FDA was somewhere between 30 and 35%, and it was felt that this fell just short given the toxicity. But when we looked at the near loop group, the response rate was 46%. The far loop was 0%. So this sort of illustrated the hazard of not really trying to tailor your therapies to the group that did best. And as a result, after a sort of fierce ODAC meeting, the drug was eventually rejected because they had not gone after the near loop specifically. Now, we've been working with another drug, sunvocertinib, that is now FDA approved, even though it's not available. And it's got a moderately higher response rate, a little better PFS, a little better toxicity. So we're making progress, but there's still wide opportunities for improvement here. All right, so let me turn now to a related kinase here, HER2. And HER2, of course, is very closely related to EGFR. Difference, it doesn't have a ligand binding. It has to dimerize for activity. And if anybody treats breast cancer here, it's often amplified. But here in lung cancer, we're dealing primarily with mutations. And if across all cancers, there's a wide variety of different mutations. This is showing the most common ones. But in lung cancer, they tend to be centralized in that red box, the exon 20 insertions. And 3 insertions, I'll call them YVMA, VC, and GSP for short, comprise about 80 to 85% of all the different insertions. So if you want a drug to treat HER2 mutant non-small cell, you need to be able to inhibit particularly the YVMA. Now, those have the same properties as EGFR exon 20 insertions, and the EGFR binding pocket and the HER2 binding pocket are very similar with some minor differences between them. And we found here from modeling that different kinase inhibitors work depending on where the mutation location was here, similar to the EGFR story. So here we're testing a set of different exon 18, 19, and 20 mutations, and we're showing the size of the pocket volume, again, with an MDS simulation. And there were a number of mutations that caused the pocket volume to shift to a smaller location. So if you see the YVMA in blue, you see that it's typically a smaller pocket volume. And that required certain drug properties that made it more challenging to drug. Now, how did we do with the existing drugs? Well, the existing drugs we had were all repurposed EGFR inhibitors, so they had a lot of wild-type EGFR toxicity. So if you look at all the EGFR inhibitors that had been repurposed for HER2, including poziotinib, median PFS 3 to 6 months and change, so not very effective here. EGFR-related toxicity is very high, a lot of grade 3 diarrhea and rash. So clearly we needed to find a different approach. So we worked with BI, and BI really had a group of wonderful chemists, and it took advantage of really small differences in the pocket between EGFR and HER2. And if you look at the pocket between EGFR and HER2, we've drawn just sort of the ATP binding pocket schematically, or the volume. The HER2 pocket's a little bit bigger. It's got an area we call the back pocket there. And it has one amino acid in particular that's different, is the 783. And so here we've overlaid the EGFR and the HER2 pockets. And if you look in gray and green, you can see the helix, the HER2 alpha C helix, sort of pushed in EGFR relative to HER2. So it's a little bigger pocket there. There's also this 783 residue that's the one residue that differs between EGFR and HER2 right in this immediate binding pocket here. And because of the location of 783 being different, it was felt that could be leveraged to try to get more specificity for HER2 versus EGFR. So in other words, if you could create a steric clash there, for EGFR, you would be able to spare EGFR activity, but take advantage of the bigger back pocket for HER2. So that led to the design of zongertinib and a whole family of compounds. So it's EGFR sparing. So on the left, this is the ratio of the mutant to wild type, HER2 mutant versus wild type EGFR. So the lower the dot, the more specific it is for HER2. And you see poziotinib and afatinib have essentially no selectivity. They had EGFR and HER2 at the same concentrations, but zongertinib is about 2 orders of magnitude more selective. So now we finally have a drug that inhibits HER2 without all the toxicity of EGFR that was really limiting us. So we did a clinical study called the BEAMIAN lung study in HER2 mutants, and we have a bunch of different cohorts. I'm happy to discuss them in detail if people are interested. But the one that was important for the FDA approval were those called the second line, people who previously had chemo, which was the standard, and then got the drug. And you saw before our prior drugs had response rate in the 30% range, median PFS 3 to 5 months. Well, here you see now we've got a 71% response rate. So really a game changer with very, very little EGFR-related toxicity, a median progression-free survival of 12 and a half months. So really much smarter drug development. So in August, this got FDA approved. And the safety profile, you remember those other drugs had grade 3 rash, and grade 3 rash is horrible, involving your whole body. Grade 3 diarrhea, you can't leave the house with. Well, here, 1 patient had grade 3 diarrhea, 0 had grade 3 rash. So, really a much better drug profile, much more active, leading to its approval. And in the first line, people who haven't had prior treatment, response rate 77. Now, chemo in this setting has a response rate about 25 to 30. So it's anticipated that this may receive an FDA approval imminently based on the voucher that the FDA gave them. Longer PFS here as well. You can see it's well north of 14 months there. And that's led to a registration study. Now, let me briefly talk about a second drug. And this is developed, actually, Matthew Meyerson and Heidi Grulich and others at the Broad were involved in developing this with Bayer. And this is a non-covalent inhibitor. So it doesn't covalently link with that cysteine here. This was also tested. Now, this was developed for both HER2 and EGFR. So it wasn't one that had the selectivity for HER2 versus EGFR, but was active in both. Also active, response rate 62%. Progression-free survival good here, 8.3 months as well. So clearly a second option. In the first-line setting, 71%, but a lot more grade 3 diarrhea and rash. So it doesn't have that specificity for EGFR versus HER2. So now we went from having no options to 2 approved options. We've also got trastuzumab deruxtecan. So how can we start using this information to tailor our therapies? Or do we just give everybody the best drug first and see what happens after that? Well, so we've made essentially every mutation that, again, is recurrent across cancers and screened them against sort of every drug that's, in the public domain, and a lot that we've sort of made here, and started looking at some of the underlying patterns of sensitivity and resistance. And the question we were asking is, can we accelerate how quickly we personalize therapies? All the work I showed you from the time of EGFR mutations being discovered in 2003 to having tailored therapy for atypical mutations was over 2 decades. The question is, can we take lessons we learned from EGFR and iterate much more quickly to identify resistance, tailor therapies more quickly, make better drugs, and really accelerate that cycle? So here we've taken clinical data from the patients that we're treating in the very first trials, literature, from what's been learned from other TKIs, and then different scanning mutagenesis approaches. We're using lentimutate here, that I'll talk about in a second, but we've also just got, we've used a couple of different scanning mutagenesis approaches. Lentimutate is sort of a quick and easy and relatively inexpensive one. It uses error-prone reverse transcriptase to randomly induce mutations. They're relatively balanced. So Ralph Kittler and his group at Southwestern have really pioneered the use of this. And I'll save a lot of data and just sort of hit some highlights here. So if we apply zongertinib and you do the lentimutate system, scan all the mutations, and then which cause resistance in the setting of a HER2 YVMA, we see a number of these mutations. Now, the cysteine at 805, remember, that's where you covalently link to the molecule. So that was very much not a surprise. But we have a number of others. The 798, this is what we call the gatekeeper mutation. And then the 783 pops up. Now, the 783 is interesting because it's a clash with 783 that gives it the HER2 versus EGFR specificity. So in other words, it occurred to us that the specificity you bought comes at a price, which opens up a new potential resistance mechanism that isn't present when you cover EGFR and HER2. So here's a patient, and sure enough, one of the first resistance mechanisms we saw is the 783. We saw 805s also, as you'd expect. Now, we also became focused on 783 because Heather was involved in the treatment of a patient, and with sequencing done with Dash Elizadeh and Max Dean, we also detected 783 there, as well as an 862 mutation. So we investigated this particular mutation further. And it's certainly the 783 and a second one, an 862, that cooperates with it, both induce resistance as well as the 805 that was expected. And we can show, that you don't suppress phosphorylation when you give the drug. Now, could we target these resistance mutations with other drugs? Well, you remember that the drug cevabertinib is non-covalent, so it's insensitive to the 805 mutation. You also recall that it's not specific for EGFR versus HER2. It doesn't leverage this 783 clash, so it doesn't care about the 783. So in fact, cevabertinib is relatively sensitive to those mutations, or those mutations are sensitive to cevabertinib. Also afatinib, again, because it hits EGFR, it doesn't leverage that clash that brings the specificity. It also is effective there. And as expected, the site where you get the covalent linkage is a source of resistance that doesn't occur with cevabertinib. And we put this in animal models, and you see in the left, that's a standard insertion, and zongertinib and cevabertinib are equally effective. But if we put in the 783 mutation, zongertinib doesn't work, but cevabertinib does there. And we can see the same with the 805 and some other mutations. So we've now taken all the drugs in the clinic and done pretty dense scanning mutagenesis as well as lentimutate and molecular modeling to essentially construct a roadmap of, if you've got this mutation, here are the set of drugs that work. If you've got this mutation, here's a different set of drugs that work. And this is on a much more rapid time scale, because where it took us 20 plus years to be personalizing things well for EGFR, these drugs just hit the clinic last year or the year before last year. So sort of real-time iterating on how we personalize while the treatment studies are going on. One other thing I'll point out, TDXD for HER2 mutant non-small cell is another drug that is FDA approved. This is an antibody drug conjugate. And we've taken a similar approach to understanding resistance there. And again, this is in collaboration with Heather and other colleagues here at Stanford. We've seen that, at least in lung cancer, the majority of resistance we see appears to be payload resistance to the antibody drug conjugate payload, and not mutation-driven resistance. We do see a little bit of resistance at the domain 4, which is where trastuzumab binds. We haven't seen that in lung cancer patients, but we've seen it in some breast cancer patients that we're able to identify. So again, non-overlapping mechanisms of resistance to an antibody drug conjugate and TKIs. And among the TKIs, they have non-overlapping mechanisms of resistance. So it starts giving us a bit of a roadmap. So right now, the current landscape is zongertinib board TDXD. Now with the approval of sevabertinib, we have a third choice. We think zongertinib is going to become a first-line therapy. And can we start tailoring which of these we use based on the specific patterns of resistance that come out? So, I hope I've sort of laid out at least a roadmap of how we can accelerate personalization there with some tangible examples. Now, in the interest of time, I'm going to sort of skip ahead of a couple of areas. I'll just sort of highlight, here's our KRAS mutant team that's really been doing wonderful work on how we target KRAS. And, what I'll say is, when we tried to study the heterogeneity of KRAS, we saw we couldn't explain it with just which alleles were mutated. So when we did unbiased clustering, we saw 3 or 4 major clusters formed. And it turns out these clusters were driven by co-occurring mutations in a set of tumor suppressors. And this really accounted for the bulk of the heterogeneity that occurred in KRAS. Well, we think this heterogeneity here, again, it's completely different than the heterogeneity you see with EGFR mutations. So here it's the things that are mutated along with the oncogene that are making the difference. What we can say is we see different drug sensitivities and vulnerabilities based on the co-mutation that happens along with KRAS. And to give some examples, if you lose CDKN2A, you adopt a mucinous TTF1 negative phenotype here. And here, a lot of this in collaboration with Monty Winslow and others, we saw that when we did CRISPR screens, STK11 and KEAP were top drivers of immunotherapy resistance. And immunotherapy, we could confirm that these drove immunotherapy resistance in patients. And then, through a more complicated analysis, we could show it actually drove sensitivity to CTLA-4 blockade and get at some of the mechanism through increased lactate secretion that this happened through. All right, what we are busy now is saying, okay, well, if it's resistant to all these different therapies, what vulnerabilities can we find? And we have identified vulnerabilities to ATR inhibition, glutamine dependence, disruption of nucleotide pools, and others. So here we're using the co-mutations to personalize therapy here as well. And I think with Lauren Byers and our small cell group, their transcriptional subgroups are driving the heterogeneity. So in the last 5 minutes, let me just talk about AI for personalizing lung cancer therapies. And we're talking with the trainees today. This was a very hot topic. And Jia Wu, who is here at Stanford, is leading a lot of our efforts there. So we've devoted the last 13 or 14 years to building a very large database that's replete with molecular data, radiomic data, outcome data, and so forth now. And using deep learning, we've been able to build a foundation model that's able to predict different outcomes for different therapies. So here, this is our immunotherapy predictor, that we can show performs far better than PD-L1 levels or other standard predictors for immunotherapy resistance. And using sort of a digital twin model with counterfactual modeling, we can say, all right, for a given patient, how would they do on chemo with immunotherapy? How would they do on immunotherapy alone? How would they do with CTLA-4? and use that to predict an optimized therapy. I'm very excited about this digital twin approach, and I think it can dramatically accelerate drug development, and I'm happy to talk about that in more detail later as well. So really now we've devoted lots of effort to building a digital twin framework that we can apply in different disease settings that can help us predict the potential outcomes and look at different treatment options and see how each of them would do as a new approach to personalizing therapies. So the bottom line from all this, different approaches are needed to tailor lung cancer therapies. I can't tell you one approach, whole exome sequencing, RNA-seq, or any one approach is going to get us where we need to be. I gave you the example for atypical EGFR mutations. It was a structure-function approach. Even within a given region, going down to the specific amino acid, we see big differences in sensitivity. For HER2, we now have been able to accelerate the time to start personalizing therapy where it's happening real time with testing these drugs in the clinic. For KRAS, to personalize it, co-occurring genomic alterations in tumor suppressors is the major factor driving heterogeneity. And for small cell, it's heterogeneity in lineage-associated transcriptional programs that define subgroups. So, moving forward, I don't think AI or anything else will get away from the need to understand biology, dig deeply in, drug sensitivities, vulnerabilities, and the hard work of developing better drugs. But I do think we're going to be able to iterate more efficiently and quickly to accelerate the time for personalized therapy and really have it concurrently occur, while we're doing drug testing in the first place. So with that, thank you for attention. Sorry for running a little late, and I'm happy to take questions.
Thank you. That was absolutely fantastic tour de force. And so we do have time for some questions, and I'm going to be scanning this side and Richard's on that side. So, okay, you guys are never shy. So, oh wait, you're second. Oh, you go online. Okay, go ahead.
Oh yeah, thank you for a great talk. So, you mentioned about the co-occurring mutation in KRAS. I wonder the impact of those kind of co-occurring tumor suppressor mutation in EGFR.
Yeah. Yeah, I think you're right, and I sort of, in a way, provided a straw man that saying, well, we need to do this in KRAS, but with the implication being it doesn't matter for EGFR. So the co-occurring genomic alterations in EGFR certainly do have an impact. So just to give a couple of obvious examples, concurrent alterations at RB and P53 predict which tumors are likely to undergo neuroendocrine transformation to small cell. P53 is associated with greater genomic instability and worse outcomes overall there. But it doesn't help us in our first pick of the first drug we're going to use. So while a lot of these are prognostic, we're not yet tailoring therapies on them. Our first tailoring is based on the specific mutation. And if you think of this hierarchically, sort of the first division is what's the mutation, and then after that, we may be able to start tailoring them on co-mutations down the road. Yeah, but I certainly hear your point.
I'm going to ask the online question, and then we will go back to the audience. Okay, so Joe Schrager, head of thoracic surgery. These are individual somatic mutations. So why not a true genetic treatment approach? We could use CRISPR if we could deliver it to go in and fix the mutations, and this could likely customize any mutation. We toyed with this approach. Thoughts?
I'd say get Nathaniel working on it here. What's up? Well, that's I just handed up to him. By all means, I think we need to think more widely about specific alterations. And I'll just give the example now for true genomic cures of hemophilia as an example by replacing, factor IX. And, we're hearing about monogenic diseases now really being cured by CRISPR. For sickle cell anemia now, this is really moving forward. So I absolutely agree with the approach. I think when you're dealing with the challenge if you're dealing with genomic cures for tumors, a typical tumor bulk for lung cancer when you start with metastatic disease is about 50 billion cells with the B. So that's on the order of 30 to 50 cubic cc's a tumor, and it's just really hard to get any therapy into every single cell. So I think it's just sort of a practical limitation, but conceptually, I think it's wonderful.
Other questions? Max.
Yeah, thank you very much for that excellent talk. I have a question regarding these digital twins and the use in the clinic. So something that is specific about oncology and digital twins is that the therapeutic landscape changes so quickly. And these digital twins obviously rely on making predictions that is based on data that we collect. But given that the landscape changes so quickly, then the data that we have actually on therapies that are relevant for these predictions is kind of scarce. And we have a lot of molecular data, so our number of variables is large and our number of sample size is small. So what's your feeling about digital twins and their use in the clinic? Do you think this will become more tractable, or do you think we'll always face this dilemma of having not enough sample size to make something very meaningful?
You had asked me that question 6 years ago, because you might have saved us a lot of time. So if you look, we worked with IBM Watson to do something called the Oncology Expert Advisor, and we spent 2 or 3 years training it, because at that time we just had chemo and a couple of targeted drugs, EGFR and ALK, and it got really good because we had about a decade of relative stasis in terms of the therapies. We didn't have a lot of changes between 2005 and 2016, 2017 that we were training on. But what happened, and if you look at the paper, it's in the Oncologist. Simon is the first author of this. We got pretty good at doing what we wanted to do then, but the problem that you just highlighted occurred because we had a decade of data sets for predicting what we wanted, and then suddenly every month new therapies started coming out, and you never had a big enough data set to be able to train it. Now, the way we're dealing with this is, and Ja feels pretty comfortable about this, there's a surprising amount of learning you can do from other drugs that you don't have to start from scratch as your foundation model gets bigger. So I'll give the example. We've got digital twins for ALK, and they were initially trained on crizotinib and then brigatinib, and we've incorporated the randomized phase 3 ALTA-1L study. It's a brigatinib versus a crizotinib there, and it performs really well there, but nobody gives any of those drugs anymore. Now everybody gives lorlatinib. Well, it looks like the work to update a functional model to just a better drug is one that you can do with a lot less data than it took to train it initially. You're training it for a little longer PFS, a little better CNS penetration. So not that it's easy, but it isn't starting from scratch. And to the extent that our drugs are incremental improvements, a PD-1 VEGF bispecific instead of a PD-1 inhibitor, a better EGFR inhibitor, or EGFR with chemo, I think we're going to be able to keep track. The challenge is, what if you have a mechanistically distinct drug? So I'll give the example of a T-cell engager like tarlatamab. Well, we've got coming up on 200 patients with tarlatamab or something, but that's still not enough to train, what we'd like. So I think when we have a new class of drugs, we're going to have to start from a further back place. But with similar mechanism drugs, we can leverage our prior learnings.
Thank you.
Great. Last, very quick question, Dr.
Smoking. You didn't mention smoking.
Smoking.
What about the
non-specific
mutations caused by smoking on top of the driving?
Yeah. So you're absolutely right. So cancer in smokers and non-smokers are two completely different diseases. They're almost as different as colorectal cancer and pancreatic cancer or something. So the genetic landscape of non-smoker cancer is much simpler. You don't tend to have overlapping tumors. Your tumor mutation burden is much lower. So they really are behaving in completely distinct ways there as well. So it does have a huge impact, and that's always one of the variables that ends up being a strong variable in our predictive models, because smoking doesn't protect you from getting an EGFR mutation. So if you're an EGFR if you have an EGFR mutant lung cancer, the rate of smoking is about what it is for the population. But those that are smokers have much worse outcomes. So you absolutely do need to take this into account.
I'm concerned about data showing stopping smoking, even if you have cancer, maybe positive, and how is that interacting?
Yeah, it's a good question. I don't have So there are specific disease settings where we know the interaction with smoking is horrible. So Billy could tell you with radiation, the outcomes are really bad when you give, especially if you're head and neck cancer, but also for lungs, smoking plus radiation, bad there. Anyway, so it's a factor we need to take into account. It's sort of a cruel irony that many of our patients are able to stop smoking for the first time when they're diagnosed with lung cancer. And I'm sure Heather's had this said many times, they'll sometimes say, boy, I wish I knew I could stop, because now it's too late, but anyway, so.
Thank you.
Okay, thank you.